Good day, good morning, good afternoon, wherever you may be. My name is Peter Girguis, and I'm on the faculty at Harvard University. And I'm very grateful to the organizers for giving me a chance to speak with you all today about life detection and about life detection on Earth and what we can learn and have learned from our efforts here to think about life detection on other ocean worlds. So I'll go ahead and share my screen here and get started with this presentation. So to me, this title is really apt in that this really isn't planet Earth. And one of my favorite quotes, which I suspect many of you have heard me say before, is that it's inappropriate to call this planet Earth when it's clearly ocean. Something that Arthur C. Clarke, the famed science fiction writer, once said, and I think really resonates with me. Now, humankind has long depended on the ocean and been humankind, frankly, has evolved around the ocean. But for millennia, humans really had no idea what lived beneath the surface. And it wasn't until the 1800s that humans set out on very deliberate expeditions to understand the deep sea, right? This enigmatic feature. And actually, these earliest oceanographic expeditions were also the first life detection expeditions. Now, new sensors and tools were at the heart of this exploration, as the scientists on board these vessels worked to develop technologies that would help them better understand the environment, these environments that were beyond our sight, our ability to just walk up to and examine. In the case of the Challenger expedition, perhaps the most famous of these, the tools you see here were designed and operated by the 6 scientists on board the expedition. And it's worth noting that these expeditions demonstrated that life was present throughout the ocean, even to the deepest ocean depths. But these were not easy expeditions, and there were many hardships that they encountered, not just the personal or logistical hardships of working at sea, but the very fact of identifying what is alive and where is it found and so on. Now, some of the challenges that they faced are still relevant to us today, even discerning life from death or telling the difference between a biological or abiotic process. They also were hard-pressed to explain some of their observations, what I call here seemingly impossible life, which is really another way of saying, how do you explain the inexplicable when you see organisms living somewhere that you don't expect them to be there? How do you go about understanding what underlies that? And then, of course, these early explorers also dealt with spatial and temporal heterogeneity. That is, you see different patterns depending on where you look. And in fact, this led to one of the greatest arguments of the latter half of the 19th century, where scientists debated the so-called azoic theory, an idea put forward by a fellow named Edward Forbes, in which he said that below about 550 meters water depth, there should be nothing that's alive. And this was a consequence of the study site that he chose and the way he chose to interpret his data, right? So the spatial and temporal heterogeneity is a real issue. Now, I will not be discussing that here today, but I did want to acknowledge that this is a big challenge we face today and will be an equally big, if not bigger, challenge in exploring other ocean worlds. But what I want to do is share just a few vignettes with you that touch upon these major challenges, these 2 major challenges, mainly number 1, dealing with life from death, and then explaining the inexplicable observations that one makes. Now, what do I mean by discerning life from death? Well, what I mean is that sometimes it is hard to really pin down what is alive. And I want to share with you a quick story here, which I'll keep brief in the interest of time. But Sir Thomas Huxley was a very renowned physiologist of the 19th century. And he described the hydrozoa and made amazing accomplishments. And in 1868, he looked at a kind of slime that he found at the bottom of preserved jars of sediments recovered from an expedition. And that slime was filled with biological sort of granules, right? He found nuclei and organelles and cell debris. And he looked at this and said, this must be the most primordial form of life there is. He named it an Urschleim. Actually, he gave it a genus and species name, Bathybius heckelei. And he said it had the essential attribute of an infinite capacity for improvement in every conceivable direction, which to me, it sounds like a kind of subtle dig. But I think what Huxley was trying to say is that this was very likely the most primitive of life forms. He saw nuclei, organelles, cell membrane fragments, and more in this slime. It was not, however, alive. And it took James Buchanan and other scientists on board the HMS Challenger to recognize that this was a precipitate formed when one added ethanol to marine sediments. And it's actually a calcium sulfate precipitate. And what he was seeing was dead cellular debris inside this slime. Now, it is cool that there was this cellular debris, but remember, Huxley's point was that this was alive by virtue of it having these attributes. And in fact, he was wrong. It's an artifact of preserving with ethanol. It's not an unreasonable conclusion for Huxley to have come to, but it was still wrong. And Huxley was very magnanimous about it and recognized the error of his conclusions, right? As we should all do in science. Now, a second example is more contemporary. In 1977, scientists discovered deep sea vents. And there, these vents were surrounded by incredibly large, very robust animals, which are not what we typically see in the deep sea. There were worms and crabs and mussels and these really beefy sort of organisms. And early on in these first few months and years, scientists hypothesized that maybe they were large because these vents were warmer and there was elevated dissolved nutrients around the vents, maybe a lack of predation, and had all these sort of explanations as to why they saw this just unbelievable amount of life around this really challenging environment. It was seemingly impossible, or at least inexplicable. But none of those explanations sufficed. And it wasn't until scientists, including Colleen Cavanaugh, Horst Feldbeck, and many others, who described this association between the chemoautotrophic bacteria and their animal hosts. Now, Riftia pachyptila is a worm that has billions of microbes per individual worm. An adult worm has more microbial symbionts living in it than there are people on Earth. And it was this symbiosis, the ability of these worms to grow microbes that could feed off of the inorganic substrates, that could harness energy from the inorganic substrates found in vents, that explains their size and predominance. So it was that critical discovery, this observation that animals can partner with microbes that can tap into these energy sources that that animals can't. It was that observation that finally helped us understand this seemingly inexplicable situation of many, many animals and very high biomass around these challenging environments. I would also note quickly that people had discovered chemoautotrophic symbioses, discovered meaning they found the animal, back in 1894 off the coast of California. They just didn't know what it was. So it was there all along. But this clam that also has symbionts, the scientists at the time simply just weren't aware of that as a possibility, right? Now, today, we continue to develop approaches to studying the density and distribution of life for microbes and animals throughout the ocean. And I wanted to share a little bit with you about some of the work we're doing that is relevant to life detection. I'll start with this idea of discerning life from death, which isn't quite accurate, but it's really about understanding what's biotic versus what's abiotic, and tell you about some of our biogeochemical studies at hydrothermal vents and how we have used those to think about discerning living versus non-living on other ocean worlds. And then I'll end with a very brief, just sort of skim of how we use geochemical maps to think about the density and distribution of life. So let's start with bioelectrochemical studies. At the end of the day, all living things have to harness energy from the environment to maintain themselves at disequilibrium. That is how we do work, and thus how we persist. And chemiosmosis is used, for example, to establish a charge gradient, right? So harnessing energy from the environment is at the heart of this. But it is also true that we are all basically bioelectrochemical machines using reduction oxidation reactions to harness energy. And we can therefore use electrochemical methods to interrogate these reduction oxidation reactions, even at the biomolecular level, as you see in the upper right-hand corner, looking at single enzymes. At the single cell level, this is a snapshot of work I've done with a colleague at MIT to isolate individual cells and electrochemically study them. And even in community-wide studies where we look at microbes that are capable of this particular metabolism, which I'll tell you about shortly. So In order to harness energy, many organisms, ourselves included, acquire substrates from the environment and bring them into the cell. We bring our electron donors and our electron acceptors into the cell. That's, in our case, aerobic respiration. Microbes can also do this anaerobically. They don't necessarily need oxygen. Some microbes do, many microbes don't. But the process of respiration is one where we use these electrons, we pull off of an electron donor, move them through biochemical pathways to an electron acceptor, and use that movement to more or less do work, right, in a very simple sense. But around 1998, or 1988 rather, scientists discovered that there were microbes that were capable of working with materials outside the cell. Eventually, this was coined extracellular electron transfer. And let me give you a simple-minded example of what this means. It means that organisms can take in electron donors, say, organic matter, right? And instead of bringing in oxygen or nitrate or sulfate to use an electron acceptor, they can actually use solid phase electron acceptors outside the cell. So iron oxides or rust are an example. So a silly analogy would be you eating something and touching your hand to a rusty plate and breathing that instead of the oxygen that you take in. Now, the ability for microbes to do this is sort of, we first started seeing it in anaerobic environments where there was a shortage of these electron acceptors, such as oxygen, of course, because it's an anoxic environment. And all the anaerobes down there are using things like nitrate or sulfate or carbon dioxide. But there are all these metal oxides, and we started realizing that microbes were capable of breathing these metal oxides. And then realized that we could actually use electrochemical systems with electrodes to actually interrogate these microbes. So in other words, if we provide a microbial community with an electrode that is at a suitable potential, something similar to a metal oxide they might see, they would actually use that as their electron acceptor, and we can measure the current and almost directly, if you will, tap into their metabolic processes, right, by measuring that electrical current. So that is, in a very simple, broad brushstroke, how we use electrochemical systems, as you see down here, to study microbial metabolism as they take their electron donors, deposit them onto the electrode, onto the anode in this case, and it moves towards an electrode of a different potential, and we measure the current. Now, we've used this to study hydrothermal vents. They're some of Earth's most extreme environments. I don't have to tell you that, vents are hot, they have low pH, and they have a lot of metals. We also now know that vents are natural electrochemical reactors. Many of the metal sulfides that are deposited are at least semiconductive or conductive. And in the inside of the vents here, if you imagine this is a vent wall, these fluids are anoxic, and outside these fluids are oxic, so you have a voltage potential and the possibility for electrical sort of current, right, for charge to move across it. And we know that microbes live inside these walls, and we know that many of them are capable of EET, this process I just mentioned of extracellular electron transfer. So we became interested in asking, if these microbes are breathing away these minerals, what role do they play in the formation of vents, right? If iron oxides are precipitating and microbes come along and reduce them and thus make them soluble, are they contributing to the growth of vents, to the dissolution of vents? How does this work? So we undertook a series of studies to look at how chimneys grow and are deposited and the role of microbes in that. In the next few slides, I'm going to very briefly tell you about some of the results. So first thing we did is we built artificial chimneys in the lab. We used high pressure, high temperature reactors. We used some ambient condition reactors, and we would take things like graphite as an electrode and subject that graphite to the kinds of geochemical conditions we see at vents. We would inoculate them with vent microbes and even add vent minerals and watch the relationship between microbes and minerals. We also did this with pyrite, taking native minerals from hydrothermal vents and basically poisoning them, electrically speaking. And we started to see that microbes and minerals were depositing, adsorbing, or relating to those electrodes. Okay, so that's, again, very brief overview, but the idea is we would recreate in the lab these electrochemical conditions that we see occurring at vents. Two very important observations. Number one, we can measure electrical current production, and as we pulse the fluids in and out of a vent, of a vent reactor with live microbes. And as we repeat that in a reactor where we have killed the microbes through gamma irradiation, there is a huge difference in the electrical current we recover. This was very exciting. The plot here shows the current density, the microamps per meter square we measured in our living and non-living reactors. So if you look at the black lines, this is microbial current production. And you can see when we inject a fresh batch of sulfide, we see that the current density stays up for a long time and eventually decays. The red lines are what happens in an abiotic reactor. You inject it, use fresh sulfide, current shoots up from abiotic reactions, and it decays fast. This is the signal of living processes. These black lines show you that the electrical current we measure is different in systems where there are living microbes than in systems without. Now, we also found that microbes influence chimney mineralogy. And again, I'm going to go through this very quickly, but we see very striking differences in the rate and mineralization in live reactors. In the presence of microbes, the metal sulfides that are deposited are primarily McKenna-white, which is very interesting as opposed to pyrite. McKenna-white is, thermodynamically slightly less favorable probably than pyrite, but it seems to be kinetically favorable. We're still trying to unpack this. We also found that even dead microbes can help influence deposition, but far less so than live microbes. And that when we start to see a chimney grow, we start to see these amalgams, these deposits of metal sulfides, building around this little electrochemical, this poised electrode, a kind of nuclei. As they build, we see that microbes are playing a key role in that, and that the community actually progresses and evolves over time, which is fascinating. So that the chimneys that we lay eyes on today are a consequence not just of abiotic, but biological processes. And we have tools to discern their relative contributions. Not just the electrical current I mentioned, but I should also tell you that vent sulfate-reducing bacteria produce iron sulfide crystals that are distinct from abiotic iron sulfides. This is work by Aude Picard that she's published in several papers over the last few years, so you could take a peek at that. So there are biological and biogeochemical consequences of EET, which I will just leave on this slide momentarily for you to read as you see fit or come back to and watch later. But the main point I want to make is that all living organisms have bioelectrochemical signatures. Work by Arpa de Bose in our lab really looked at how to tease those apart using some particular cyclic voltammetric approaches. And again, we can see these in living cells and compare it to dead cells. And these electrochemical studies actually underscore this inextricable relationship between biological processes and the abiotic environment. But the electrochemical approaches allow us to see the relative contributions of both. As of late, we're doing work led by Izzy Baker and Bridget Conley, two students at Harvard and University of Minnesota, showing that the capacity for EET is in fact widespread among the bacteria. And this paper is in review right now. So if you have any questions about it, let me know and I'll happily answer them. But bioelectrochemical systems can be deployed on ocean worlds, our own and those beyond. Now, we deploy these in our ocean to make these wireless seafloor sensor networks, basically harvesting energy from the seafloor, from seafloor microbial metabolic processes to power sensors to study the seafloor. So that's very cool. But working with Colin Bowie at MIT, we've also built and used systems as small as a microscope slide to study these processes of EET and frankly, just electrochemically interrogating organisms at a microscale. Now, in just the last minute, I said I'd be brief here, but I wanted to talk about how we can also use geochemical maps to better understand life. And this is something that as we explore ocean worlds, we may have an opportunity to do. How do you go from geochemical measurements to understanding the density or distribution of life, right? Now, there's lots of different ways you can imagine using sensors to look for the presence of life. But one thing I think is equally important is to be able to use these sensors to make a geo-referenced geochemical map, and then to start asking where are the sites where life is most likely to be. We've developed and used this underwater mass spec for about 15 years now to go down to the seafloor and quantify dissolved volatiles over time. Working in the deep sea has made it hard. It's hard to study geochemistry sometimes, especially if you're talking about gases, because you have to collect a water sample, historically, just collect a sample, bring it to the surface and work on it. But the mass spec allows us to make these inquiries in real time on the seafloor. And so my brief snapshot of what this might look like is shown here. We use our mass spec to go to places like seafloor vents and seafloor seeps and even the oligotrophic waters of the deep Atlantic and Pacific, and make these geochemical maps. We use the mass spec to sip water at different points, to look at oxygen, nitrogen, sulfide, CO2, hydrogen if it's present, methane if it's present. And we make these maps in basically a GIS framework. We can then superimpose upon that the distribution of the animals we see and start to think about the relationship between energy availability in an environment and the abundance of biomass in that environment. And this gets very interesting and very complicated very quick as we start to see how matter and energy flow from the lithosphere here on Earth into the biosphere through microbes. And we have about 14 papers or so on this with many wonderful collaborators that have employed this kind of in situ geochemical mapping to understand that relationship. So I'll just end by saying that here we are in the 21st century, and we're still coping with some of the challenges that our colleagues did in the 19th century. But through newer technologies and frankly, thoughtful experimentation, we're developing exciting new ways to do these studies. This is a picture of an abyss lander. This lander developed in my lab with support from NASA that allows us to take so many of the instruments we use in the lab and put them on the seafloor and do studies over time to use that temporal variability to understand microbial processes. But the challenges of ocean exploration are similar on Earth or beyond. And I would strongly say that and advocate for more crosstalk among oceanographers, space scientists, and industry experts to get us to where we want to go. And of course, I have the fortune of sharing this fantastic work, a lot of which was done by members of the lab shown here in our holiday card, our Star Wars holiday card. I also want to thank the funding agencies, NSF, NASA, the Moore Foundation, the Lindbergh Foundation, and ARPA-E, as well as Harvard University for their support. And I would like to thank you very much for your time, and I really appreciate you listening to this talk. Be well. Bye-bye.